Hello, everyone. Welcome to this short introduction to machine learning and artificial intelligence in the Center for Advanced Bioenergy and Bioproducts Innovation, or CABBI. I'm Saurabh Sinha, a professor in computer science and the Carl Rose Institute for Genomic Biology at the University of Illinois. I'll begin with the highest level question one can ask in our context. What exactly is machine learning? Well, if you look up Wikipedia, you'll find that it's the study of algorithms that automatically improve through experience. So then you'll wonder, what is experience for an algorithm? And the answer is simple. It's the algorithm seeing more and more data. And the next question will be, well, what does it mean for an algorithm to improve? Improve what? The answer to that is the algorithm's ability to make predictions from data. All right, so now that we've clarified the direct meaning of that definition, let's ask ourselves, what sort of predictions are we talking about here? Well, one type of prediction that we're interested in making within our center's missions is that of gene expression in a particular biological or cellular context. A gene's expression level in a condition may be predictable if we know the expression levels of its regulators. Machine learning algorithms can perform such predictions. Another kind of predictive ability of interest to us is that of the yield of a metabolic pathway. Such pathway yields can be accurately predicted if we know the levels of the key enzymes in the pathway. Again, machine learning algorithms could make such predictions. Okay, then a natural question is, do we need to teach the algorithm all about molecular biology and or biochemistry if it has to make the kinds of predictions I just mentioned? Thankfully, the answer is no. These algorithms are rather general purpose, instead of being overly specific to the domain their predictions belong to. And the key idea underlying their workings is shown in this simple plot, which you are likely familiar with. Let's say the algorithm has to predict a pathway's yield based on the level of a particular enzyme. This is an oversimplification, of course, but it makes the point. Suppose we have data on the pathway yield at different levels of the enzyme's concentration. The algorithm will then try to find the trend inherent in the data, in this case, a straight line that indicates that any given increase in the enzyme's concentration will result in a proportional increase in the pathway's yield. Once the algorithm detects such a trend or mathematical function mapping the input variables, i.e. the enzyme levels, to the output variable, i.e. the pathway yield, it is ready to make predictions for any given enzyme level or combination of enzyme levels in the future. At this point, I often see skeptical faces in the audience, and the unsaid sentiment is, all this is fine, but it's too naive to work for biology. And my response is, it's not, because it's been shown time and again to actually work. There are tens of thousands of researchers demonstrating the power of machine learning for making biological predictions. And the reason it works, despite my slide appearing overly simplistic, is that in reality, the learned functions can be fairly complex. For instance, they need not be linear functions, and they may be functions of more than one, even thousands of variables. I'll change gears now and present a very specific way in which machine learning is being used in the CABBI Center today. The example involves our attempt to understand gene regulatory mechanisms underlying stress tolerance in the yeast strain Escherichia orientalis. This is collaboration between my lab and the labs of the Yoshikuni and Maslov labs. The data we have is a matrix of gene expression values under varying stress conditions. So rows in this matrix represent genes, and the columns represent biological conditions. And let's say we want to understand the regulation of a particular gene, G, whose row is shown here at the bottom. Now, We know that the gene G is likely regulated by one or more transcription factors, so we focus on the expression levels of all the transcription factor genes. Let's say there's n of them, also available from the same data matrix. What we do here then is to train a machine learning model to predict the expression level of gene G in a given condition from the levels of all the TFs in that condition. Once such a model is trained, we can interrogate it to find out which transcription factors are the most useful variables for its predictive ability, and that gives us candidate regulators of gene G. Continuing the same example, in another project, we're using a convolutional neural network, which is a specific type of machine learning algorithm, to predict a gene's expression from not only the expression levels of the transcription factors, but also some information on the mechanisms by which those transcription factors might act. In particular, this machine learning model utilizes the gene's promoter sequence and probabilistic descriptions of the DNA binding preferences of each transcription factor as additional data beyond just the transcription factors' concentrations in varying conditions to predict gene expression. In this example, we saw how different types of information about a gene, for example, its promoter sequence or a cellular condition, or information about a cellular condition, such as levels of transcription factors in that condition, can be used to predict the gene's expression in that condition. So generalizing from that example, we ask, what sort of data sets are fair game for machine learning algorithms in our center's context? And while the answer depends on our specific biological goals, a general view is that the data is a collection of measurements on a variety of things, for lack of a better term, in a variety of conditions. And there's measurements on some other thing in those conditions, or a yes-no property of something in those conditions. And the task is to predict this latter thing from the other measured things. This is a very high-level description of a machine learning task, and some important considerations related to such tasks include the need for measuring in as many conditions as possible. Ultimately, the machine learning algorithm is trying to learn to predict something in an unseen condition. So the more, or rather more diverse the conditions that it has already seen during training, the better equipped it will be in making predictions. One thing that happens frequently in biological applications of machine learning is that the algorithm tries to make predictions based on a large number of variables and ends up being rather complex and hard to interpret. This is well recognized in the community, and one of the major goals of the day is to make machine learning algorithms and models more interpretable and less complex. An important thing to remember when talking about machine learning algorithms and predictions is that the algorithm must predict correctly on unseen data that it gets in the future, not just the data that it saw when it was being trained. This is called generalizability of the machine learning model and is an important criterion for when training these models. Biological data sets are routinely obtained from a combination of different technologies, such as DNA sequencing, transcriptomics, epigenomics, metabolomics, etc. And while the machine learning algorithm typically sees all these different types of data ultimately as numbers, those numbers clearly mean different things. And the algorithm is probably better off respecting the different semantics of heterogeneous data. And so the right way to handle this heterogeneity of measurements available in the data sets is also an important consideration in machine learning for biology. Another big realization from years of experience applying machine learning to biology is that prior knowledge matters. A data set, such as the one shown here, can be used to train the machine learning algorithm, and the algorithm will do its best given the data. But it almost always helps if the algorithm is additionally provided information specific to the system we're trying to understand. For example, known relationships and functions of genes, if those are measurements of genes, protein-protein interactions, pathway structures, if those are pathway yields, and so on. If you're a student watching this video and the prospect of applying machine learning to biological problems excites you, you might be wondering how you can get started. The good news is that there's a variety of courses and certificates available today for training in machine learning at different levels of experience and expertise. These courses are offered by a number of different sources, including our computer science department, where I belong, as well as programs in data science and information science, statistics programs, etc. Often, the prerequisite for going on to this step of training is simply the ability to code and some basic familiarity with statistics. The machine learning training builds off of those core skills. That's all I have in this short tutorial. All the best, and thank you.